be talking about all the material post midterm 2 from now. Thanks for coming back. Make sure you sign in using the link below, and like usual, comment down below if you have any questions, and we're gonna get right into it. Okay, so contributors to genetic variation, right? We started talking about mitosis and meiosis, right? And meiosis is producing the unique daughter cells that are the sex cells that are then combined to create a zygote and thus a human being, right? So, like, genetic diversity in this process results in genetic diversity in our overall human gene pool. So, what are those things that are making us diverse, right? So, like I mentioned before, you have crossing over, which is in uh, in metaphase right here. When those chromosomes line up, when those homologous chromosomes line up in the middle, you have crossing over occur, which is when a segment from one of the homologous chromosomes switches with another homologous chromosome. So then that promotes genetic diversity in that the two homologous chromosomes that are then splitting apart um, will not be completely identical to your father's or your mother's chromosome, right? Then independent assortment, which is the concept that when you have your homologous chromosome line up on metaphase plate, obviously in this image, it is showing that there's one chromosome, but we know there are several more. So when they line up, they're lining up independent of each other. So it's not to say that all the mother's chromosomes are lining up on this side and all the father's chromosomes are lining up on that side. So that again promotes genetic diversity. And then random fertilization. So like when we're talking about these two, we're kind of talking about processes within meiosis that cause genetic variation, but random fertilization is the concept that these sex cells that are produced, so your sperm and your egg cells, the ones that meet and form a zygote are completely random. Um, though choosing which ones out of the millions of sperm cells and the eggs that you do have is completely random. So it's referring to that phenomenon. So these are the three major um, processes or attributes to genetic variation. So moving on, then we talk about a karyotype, and this is an example of a karyotype. I believe this is the human karyotype. So, as you can see, the human karyotype is a depiction of chromosomes, and they're organized by their size, their shape, and their staining pattern, right? So it just gives you an overall image of all the DNA in an organism's gene pool, if you understand what I'm saying. Okay, so what are the steps that you need to take to obtain a karyotype? So, to start with, first of all, you gotta obtain a sample. So, whatever it may be, if you're looking for a karyotype of a plant or of a human, you would have to obtain a sample of that DNA, right? And then you're gonna culture cells, and this is basically when you have a culture, you're often swabbing it on a plate, and each single colony that's on that plate is now completely individual and has completely quote unquote pure DNA because that colony of cells was formed by one original cell. So through mitotic division, all those cells have the same DNA. So you're getting a really pure sample. And then from there, you're going to stop division at a metaphase. Um, why metaphase, right? So if we kind of go back here, this is metaphase where your chromosomes are lined up in the middle. And you would want to stop it there because your chromosomes have already paired themselves up. It's a really convenient location to stop it, right? You've done part of the work already by stopping it at metaphase. And then you stain the chromosomes as shown right here, and then you examine and organize them into a very nice graphic as shown here. And that's how you obtain a karyotype. Um, Next thing we talk about is alterations in chromosomes, and I've kind of organized it here so that you can differentiate the different types of alterations. So there's two different types of alterations. You can get an alteration in number and an alteration in structure. Let's talk about number first. You have aneuploidy and polyploidy. So, aneuploidy is what we think of when we think of genetic disorders, right? So, it's when you have one extra or one um, less chromosome. So, it's not referring to the overall set, it's referring to just one of the chromosomes. So, if you have a trisomy, which means you have three of one chromosome, and in this case, chromosome 21, I believe, the offspring is going to have Down syndrome, right? But if you have a monosomy, which is having one chromosome instead of the two that is expected of a human set of chromosomes, because we're 2N, you're going to result in Turner syndrome. So that's an example of aneuploidy, where you have one extra chromosome, and this is because of non-disjunction. And I'm going to go back to our image right here. Non-disjunction would be when you have metaphase, and you have an issue in the division of these cells, right? In anaphase, they pull apart. So if non-disjunction would be if these two chromosomes went to one side, leaving this side with none, or if right here these two sister chromatid went on one side, leaving this side with none, right? So you would have an uneven splitting of your homologous chromosomes or your sister chromatid. So that's non-disjunction, and that results in this change in one chromosome in your overall set of chromosomes. Polyploidy refers to your overall set, and it's having more than two sets of chromosomes. So this isn't necessarily a genetic disorder that we see in humans. You don't really see humans with like a set of 3N, but what you do see is you see different types of organisms, like strawberries and bananas, that have more N, and I'm sure you guys could list some really unique, interesting factors in the comments below. So that's alterations in numbers. So we're going to talk about alteration in structure. Way to explain the structural alterations in chromosomes is to kind of look at it visually. So I've included this very classic image that explains it perfectly. So deletion is basically backspacing, right? So you have D in the original chromosome, and it's just gone in B. It was, or in the altered chromosome, it's like completely gone. It's not there anymore. It's deleted. And then in duplication, you have your B and C here in the original chromosome, and then it's just copy pasted right next to it in duplication. So again, very straightforward, duplicated, right? The next one is inversion, which is kind of not as straightforward. Yes, inversion means you flip it around, so instead of going BCD, you're now going DCB. But it's important to understand that you're not talking about this going A, B, C, D, E, F, G, H, and then this going A, B, C, D, E, F, G, H, right? You're just talking about a segment of it, because if you were talking about the whole thing, then nothing really changed, right? You still have the same sequence, you still have the same connectivity. But in this case, it's just a segment of that DNA that's changing, so you do have a different sequence. Instead of having A, B, C, D, E, you have A, D, C, B, E. So that's inversion. The next one is reciprocal translocation, or just translocation, right? And this is when a segment of one piece of a chromosome is swapped out with the segment of another piece of chromosome. So this AB right here is now on this chromosome, and MNO is now on this chromosome. So pretty straightforward. Just keep in mind with inversion, you're talking about a segment and not the whole thing, but that covers the structural alterations in chromosomes, and we talked about the difference in numbers, um, like the alterations in chromosomes. So the next thing are the experiments. I'm gonna again stop right here, and I will meet up with you guys in a different video. Okay, don't forget to sign in and comment your questions.